In this project, we aim to understand how Ebola virus affects the human body's immune response. Ebola virus, or EBOV, is one of 3 genera of the Filoviridae family and has 5 major species, named for their geographical origins: Weston, Budamungo, Sudan, Thai Forest, and Zaire, which is now known as the Democratic Republic of the Congo. EBOV is an incredibly deadly virus which causes Ebola virus disease, or EVD. Ebola virus disease has a mortality rate estimated between 25 and 95%, depending on the disease progression. EBOV, like other viruses in the Filoviridae family, manipulates the host's innate immune response, which overloads the immune system and causes serious and systemic complications, such as hemorrhagic fever and septic shock. EBOV is especially severe because of its rapid effect on a wide array of tissues, including the liver, spleen, kidneys, and blood vessels. Ebola virus is a membrane-enveloped filamentous, meaning thin, resembling a filament or a thread, negative sense single-stranded RNA, which is converted into mRNA upon cellular entry. It has only 7 genes which code for 8 proteins. Understanding how Ebola virus hijacks the immune system is central to understanding how to design an effective vaccine and therapeutic treatments for the virus. This project aims to understand the ways in which EBOV affects the human immune response, an area of inquiry which is receiving focused attention, given the especially dangerous nature of filoviruses in general, and Ebola virus in particular. To answer our broader question, we pose the following constitutive inquiries, which will be subsequently answered in this presentation to arrive at our answer. First, how does inhibiting methylation affect EBOV virulence? Second, what are the methods by which EBOV can be safely studied without biosafety level BSL-4 containment? Third, are there therapeutics or vaccines being developed to contain the spread of EBOV? And finally, what are the lasting effects of EBOV infection on the body?
In this presentation, we are going to discuss the relevant biochemical pathways of the Ebola virus, or EBOV, as well as give a history of its global burden. We will then discuss the virus's pathogenesis and give an overview of the immune system before getting into some specific immune responses to an EBOV infection. We will describe the methylation cycle and why it is important to viral RNA, as well as detail a possible synthetic antiviral drug which is capable of inhibiting said cycle. The results of both of the main papers used in this study, one regarding T-cell responses and the other regarding natural killer cells, will be explained. Penultimately, we will present on safer methods for studying the virus, as well as a possible vaccine. And finally, we will detail how the Ebola virus is able to evade the immune system and propagate.
So in this presentation, we're going to cover 3 biochemical pathways, and they're interspersed as a general introduction into a biochemical concept or research related to Ebola virus.
The epidemiology of a virus can be impacted by geographical, sociopolitical, and genomic factors, which affect who gets it and where the virus is most prevalent. While it is widely thought that EBOV originates from small mammals such as bats because of the similarities in disease progression in mammals, there is as of yet no definitive evidence for this. EBOV outbreaks have been most common in Central and West Africa. EBOV affects all human beings regardless of age, gender, or race. However, EBOV tends to affect isolated rural communities first because of the greater exposure to animals in the wild through the hunting of bushmeat. The geographic spread of the disease is usually not severe, but the impacts of the outbreak within a concentrated geographical radius can be serious. Transmission of EBOV occurs primarily through direct contact with bodily fluids. While there is no evidence of airborne transmission, it is always possible that such a rapidly mutating virus such as EBOV could potentially generate the capability for airborne transmission, which is supported by the fact that studies have shown that EBOV and aerosolized droplets can be infectious. Secondary cases through indirect exposure are strongly correlated with caregiving, burial, and other domestic responsibilities. In addition to geographic and sociopolitical factors, the epidemiology of a virus can be shaped from a genomic standpoint. The actual genetic material of the virus can change and mutate into additional species, something that has occurred frequently in the Ebola virus and which makes tracking the spread of the virus very difficult. Genomic data enables a clear understanding of which EBOV species is causing an outbreak and the disease differences between virus species. Importantly for EBOV, the glycoprotein or GP gene, which enables viral entry into the cell, has one of the highest levels of genetic amino acid diversity. One important change is the substitution of a valine for an alanine residue, which has the potential to change the interaction between the glycoprotein and its host receptor. This suggests that the gene for the glycoprotein is subject to high amounts of mutation, which affects host-receptor interactions, an important factor when considering viral spread because of the glycoprotein's role in enabling the virus to enter the cell. Vaccines are usually developed to trigger the body to produce antibodies that can recognize proteins on the outside of a viral cell, like EBOV glycoprotein. If this protein is mutating and changing between species, one vaccine may not be able to protect against all EBOV species. The pathogenesis of EBOV is marked by the simultaneous suppression and overactivation of the host immune response, resulting in the failure to activate essential effector cell functions, leading to uncontrolled virus replication, systemic virus dissemination, and inflammation and tissue damage. The details of these processes will be discussed later on. After the virus invades the cell through the combined efforts of viral protein 40 and glycoprotein, EBOV targets dendritic cells and other monocyte macrophages. Viral viruses like EBOV readily replicate in these cell types, causing them to die and spreading huge numbers of viral particles to the extracellular fluid. Dendritic cells are primarily responsible for initiating the adaptive immune response. Targeting these cells also inhibits the ability of the infected organism to produce antibodies against it, suggesting the possibility for reinfection and posing a particular challenge to vaccine development. Part of what makes Ebola virus so deadly is its capacity to quickly spread to many different areas and tissues within the body. In the first 3 days after the onset of symptoms, the viral load in the blood increases exponentially to over 10 to the 5th viral particles per milliliter, indicating the host innate immune system is overcome. Following sudden onset fever and malaise, if Ebola virus disease advances to cause hemorrhagic symptoms, the risk of mortality increases to 30 to 50%. If Ebola virus disease progresses further to multi-organ dysfunction, mortality can be up to 90%. Infection of crucial organs, such as the liver and spleen, occurs early in disease progression. Tissues of deceased animals show EBOV infection in endothelial cells, fibroblasts, hepatocytes, adrenal cortical cells, and epithelial cells. Without medical care, death usually occurs within 10 days of infection with EBOV. Ebola virus was first reported in 1976 in the Democratic Republic of the Congo, then known as Zaire, with an outbreak over 2 months generating 318 cases and a mortality rate of 88%. From 1977 to 2014, there were 12 smaller outbreaks in Middle Africa, with case counts ranging from 32 to 315, and mortality rates of 47% to 89%. The 2013 to 2016 outbreak was the first in West Africa and has similar disease characteristics as previous outbreaks, with a mortality rate around 70% and a reproductive rate between 1.5 and 2.5. During this outbreak, there were roughly 30,000 cases with over 11,000 deaths. The 2013-2016 outbreak of Ebola virus in West Africa originated via human contact with an animal, probably a fruit bat, in the small village of Meliando in Gwenkado Prefecture, Guinea. The 2013-2016 epidemic was the first in West Africa and spread from Guinea to Liberia, Sierra Leone, Nigeria, Senegal, and Mali. In Guinea, Liberia, and Sierra Leone, the spread of infection was especially severe. Approximately 60% of healthcare workers who responded to the pandemic were killed by EBOV infection. There have been 2 recent outbreaks in the Democratic Republic of the Congo, both reported in 2018. The outbreak was announced as over on July 25, 2020, after 3,470 confirmed cases and 2,287 deaths, which is a 66% mortality rate. Just a few months ago on June 1st, another outbreak was announced in Mbandaka, Equateur-Provence of the Democratic Republic of the Congo. As of July 25th, there were 63 confirmed cases and 27 deaths.
We're going to do a quick overview of the immune system. The immune system can be split into 2 parts, the innate and the adaptive. Despite this dichotomy, however, I want to emphasize that all areas of the immune system are intricately interconnected and shouldn't actually be viewed separately. And as the name implies, the innate immune system is a non-specific general response that serves as a sort of first attempt at cleansing the body of our invaders, hereby termed pathogens. The innate immune system is composed of the physical barriers of our body with which we are familiar, such as the skin and hairs that we all possess. These serve to physically keep out the pathogen. Moreover, there is the blood-borne component of the innate immune system. At the bottom left, we have our phagosomes, which are our neutrophils, eosinophils, basophils, and macrophages, that engulf and digest the pathogen in a process called phagocytosis. In the middle, we have antigen presentation, which is when a dendritic cell or a mast cell captures a portion of a pathogen called the antigen and displays it on its own surface for certain other immune cells to recognize. And upon recognition, these cells can be activated to perform more functions. At the bottom right, we have our cytotoxic response by our natural killer cells, which get rid of already infected body cells. And lastly, the complement cascade, as depicted at the top, helps to augment actions undertaken. The adaptive immune system has fewer moving parts and is a more specific response geared towards specific pathogens. If we are infected by a pathogen a second time, it is thanks to the adaptive immune system that we can get rid of the foreign body so quickly. This is because of two kinds of immunity. Humoral immunity is related to antibodies that were produced in the first infection still being maintained in body fluids. So when the second infection comes, the immune system recognizes the repeat invader. The other immunity is cell-mediated immunity, which is the amplification of the immune response through recruiting and activating other cells upon antigen recognition. There are two cells involved, B cells and T cells. B cells come from the bone marrow and use antibodies, which are Y-shaped proteins that have a variable section in their sequences. Each B cell has a unique antibody, so there's a really good chance that some B cell will be able to bind to a new pathogen. And upon binding, the B cell is activated and begins to divide and differentiate into B cells that can go on to do different jobs related to humoral immunity. T cells do not use pathogens and rely on antigen presentation to become activated. Once activated, it can also differentiate into different T cells that do different jobs related to cell-mediated immunity. So, the cytokine storm is an abnormal version of the immune response. There's a lot of different kinds of cytokines, which can be proteins or peptides, that serve as signaling molecules for the immune system and can be produced by a number of cells. Cytokines can be pro-inflammatory in that they stimulate inflammation, and when you produce too many of these molecules, the overinflammation can lead to multiple organ dysfunction that can be fatal. Ebola virus have a certain component called glycoproteins that they shed after invasion. These glycoproteins can float around and go on to bind onto the toll-like receptors of dendritic cells and macrophages. This stimulates the cell to release a lot of cytokines that go on to activate a number of other immune players. And when this is overdone as a result of a lot of infection, a cytokine storm occurs that can be dangerous for the patient. Methionine is an essential amino acid that can help to generate a number of important biomolecules for our bodies. As we can see in our figure, methionine adenosyl transferase can generate S-adenosylmethionine, or SAM, which is one of the very familiar one-carbon unit carrying cofactors we've seen. The reason that the SAM is such a good methyl donor is because the adenosine group destabilizes the sulfur-methyl bond, since the sulfur is positively charged after losing its lone pairs to make SAM. Something else that is really important to note is the fact that S-adenosylhomocysteine, or SAH, which is made from SAM, is a negative feedback inhibitor. More specifically, at higher concentrations, SAH will inhibit the methyltransferase enzymes, and so if there's a way to inhibit the SAH hydrolase enzyme that uses the substrate, there will be a buildup of SAH, and therefore inhibition of methyltransferase enzymes involved in biological processes of pathogens, such as 5' capping of RNA and post-transcriptional modification. As such, this is the case for the drug we will be discussing, L-3-dezainaplanacin.
The Ebola virus hides itself from the human immune system. As a consequence, the virus replicates exponentially before the immune system can mount an attack against it. Since so little is known about Ebola's mechanism of action, researchers have turned their attention to broad-spectrum antiviral agents. One such potential drug is L-like 3-deazinaplanacin, or L3-DP. The synthesis of L3-DP proceeds via several steps from a simple precursor. In the first step, dry butyl lithium is used to deprotonate a terminal methyl group on tert-butyl methyl ether. The deprotonated methyl group is then allowed to attack the ketone on the precursor molecule to form a newly substituted cyclopentene molecule. After a protection step, this second precursor participates in a Suji-Trost reaction, whereby a palladium-2 atom, stabilized by bound chlorine and nitrogen atoms, is able to complex itself to the delocalized pi orbital in the cyclopentene ring. This complex expels the protected oxygen substituent, which can then re-attack the cyclopentene ring on the other side of the palladium complex. The complex dissociates from the molecule, whereby the resulting precursor has flipped substituents. After a deprotection step and a protonation event of the newly deprotected hydroxyl group, The intermediate participates in a Mitsunobu reaction, which prepares the 3-deazoadenine Bok2 to attack the proximal side of the carbon substituted with water. In the Mitsunobu reaction, triphenylphosphine is activated using diisopropyl azdecarboxylate, or DIAD, to replace water on the cyclopentene ring with the diazobok group formerly attached to ribose. The resulting product is treated with concentrated hydrochloric acid to hydrolyze the Bok2 groups as well as the isopropyl group, protecting 2 of the remaining hydroxyl groups attached to the cyclopentene ring. The acid similarly hydrolyzes the tert-butyl ether on the alkane substituent. The resulting molecule is L3DP. The reason that L3DP is important in inhibiting Ebola's action is because it can inhibit the function of acetinosyl L-homocysteine hydrolase, or SAHH. SAHH works by employing NAD+ to hydrolyze acetinosyl homocysteine, or SAH, to L-homocysteine, HCY, and adenosine, ADO. HCY is used in a cycle to generate acetinosyl L-methionine, SAM, a widely used methyl donor necessary for the proper functioning of L polymerase. The process which generates SAM is known as the activated methylation cycle. The activated methylation cycle is a necessary part of viral transcription. L3DP is a competitive inhibitor which might prevent the entry of SAH by doubly protonating the active site bases of SAHH. L3DP may also contribute to induced oxidative stress by allowing for NAD+ to become irreversibly reduced to NADH.
As we have discussed on previous slides, Ebola is an incredibly fatal disease. What is less known is that for those who are fortunate enough to survive, will often experience harsh symptoms for months or even years after a detectable virus has cleared. These symptoms include some more manageable ones, such as headaches and fatigue, but there are a lot more debilitating ones, such as muscle pain, joint pain, and ocular symptoms. This is known as post-Ebola syndrome, or PES. In hopes of getting to the root of PES, the authors of this paper surveyed survivors of Ebola every 6 months. Patients were classified as having PES if they reported symptoms like the ones I mentioned earlier at the first screening. The authors did this as a series of assays that assessed the presence or absence of specific compounds related to the immune system in patients by doing mainly blood tests, as well as more specific ones that tested for things like immunoglobulin. The authors provided 3 possible explanations for their results, of which are tabulated on this slide. In the interest of time, I'm only going to touch upon 2. The first is that the virus may actually still be present in patients with PES, despite it not being detectable. The authors deduced this from the fact they found elevated T-cell responses in patients with PES, specifically CD8+ and CD4+, of which are responsible for mediating the adaptive immunity of cells and shaping the immune system's response. Additionally, they found elevated interleukin-13 in PES patients. IL-13 is a cytokine that is secreted by CD4+ T cells, so the increased level is directly related to the fact there is already higher CD4+ T cell activation in those with PES. It is difficult to assess the viral load in specific areas of the body that don't have a lot of immune surveillance, such as joints and eyes, due to the invasive nature of those assays. This would not only explain some of the symptoms, but it would explain the elevated T-cell responses. Another idea they had was that Ebola has triggered an autoimmune disease resulting in tissue damage. This idea is backed up by the fact there were heightened rheumatoid factor levels in patients with PES. RF are proteins produced by the immune system that actually can attack healthy tissues. Because RF is associated with other autoimmune diseases, this is a very viable explanation.
This study investigated the role played by natural killer cells in the innate immune response to Ebola virus infection in a highly relevant ex vivo model of human peripheral blood mononuclear cells, both before and after vaccination. Natural killer cells are activated in the innate immune response, as opposed to in the adaptive immune response, by cytokines and viral ligands to kill virus-infected cells. Previous studies have indicated the potential role natural killer cells play in the immune response post-vaccination for other viruses as well as for Ebola virus. In this study, the authors wanted to know what role natural killer cells play in responding to Ebola virus vaccination. They were especially interested in looking at the mechanism of natural killer cells in response to Ebola virus glycoprotein, which is targeted in vaccine development because it is essential for cellular invasion, is highly immunogenic, meaning it's able to induce a humoral and/or cell-mediated immune response, and is the only protein exposed on the surface of the mature viral cell. As their main model, they used peripheral blood mononuclear cells isolated from human subjects before and after vaccination with a novel 2-dose homologous Ebola virus vaccine, which has been proven to be safe and effective and is currently in phase 2 and 3 clinical trials. In addition, they treated some of the ex vivo samples in vitro with soluble Ebola virus glycoprotein. They had 3 major findings. First, vaccination induced proliferation and activation of less differentiated natural killer cell subsets. Second, stimulation of peripheral blood mononuclear cells with Ebola virus glycoprotein induced TLR4-dependent secretion of high concentrations of inflammatory cytokines, mostly from CD14+ monocytes and accessory cell-dependent natural killer cell activation. Finally, Ebola virus glycoprotein-induced natural killer cell activation was inhibited by neutralizing antibodies to IL-18 and IL-12 and was enhanced by IL-10 receptor blockade. In summary, this paper characterized the natural killer cell response to the novel 2-dose vaccination regimen and demonstrated that the TLR4 receptor and innate cytokine response to Ebola virus glycoprotein stimulate and regulate the natural killer cell effector response. Ebola virus remains a public health threat, and with a mortality rate several times that of COVID-19, the scope of a widespread outbreak would be extremely devastating. A vaccine is clearly desirable, and previous studies have shown a promising role for natural killer cells in early vaccine-induced immune responses. Wagstaff et al. Were able to characterize the immune response induced by Ebola virus vaccines and demonstrate innate cytokine responses as a result of Ebola glycoprotein were integral to inducing and regulating natural killer cell function following vaccination.
One of the main hurdles that scientists face when studying Ebola is the containment level needed in order to safely investigate Ebola. Not only is it expensive to create facilities that enable the safe research of Ebola, the researchers themselves are always at risk. The method I'm about to discuss eliminates a large amount of these risks. Simply put, let's say you're trying to study a G protein in Ebola. If you were to do this, it would require Ebola cells, i.e. live virus. But what if you could simply take the gene encoding the Ebola G protein and put it into another virus cell that is less dangerous? This would enable you to eliminate a lot of the required facilities. This is exactly what happens when one creates recombinant single-cycle infectious vesicular stomatous virus, VSV. VSV functions as the host virus for the encoding genes. Like Ebola, it is a negative strand RNA virus. The viral entry of VSV is quite similar to Ebola, as you can see in the figure on the slide. I'm now going to discuss 2 applications of this method that pertain to the papers that have been previously discussed in this presentation. The authors of the first paper assessed how patients responded to Ebola antigens. They were able to produce these antigens by creating a recombinant VSV, rVSV, which encoded the Ebola genes for a number of essential proteins of Ebola. The figure below describes the process in which these rVSV were created. First, the Ebola genes were amplified and inserted into plasma vectors. Then, using reverse genetics, they were plaque purified, meaning that only the plasmas with the Ebola genes were further studied. The RNA from the cells that contained the rVSV were reverse transcribed into cDNA and amplified. Then the protein expression levels of the cells were analyzed with Western blotting using an anti-flag antibody. The authors of the second paper touch upon how rVSVs have been used to develop vaccines. VSV can be used to create a vaccine due to the fact the virus itself is rare, so only a small portion of the population is immune to VSV. The vaccine is known as rVSV-Zaire, which prevents Ebola caused by Zaire strain of the virus. This vaccine was developed by replacing the G protein of VSV with Zaire strain G protein. This vaccine only protects against this specific strain of Ebola. There have been attempts to develop vaccines that protect against multiple strains, but they have not been fruitful. The vaccine itself was developed at the Public Health Agency of Canada and distributed by Merck, located in the Boston area, and is a pharmaceutical company. Not only has the vaccine been patented, it was FDA approved in 2019 and awarded pre-qualification status by the World Health Organization. It has recently been approved for use in 4 African countries. The successful production of this vaccine highlights the impact of this method on the study of Ebola.
The Ebola virus, or EBOV, is capable of infecting humans and causing Ebola virus disease, or EVD. EBOV is able to incapacitate both the innate and adaptive immune responses. Much of the mechanism by which the EBOV evades the immune system remains unknown. However, it is clear that EBOV manipulates an innate immune deregulation in its quest for viral propagation. After entry into the host bloodstream, EBOV uses its glycoproteins, or GPs, to gain entry into host cells. Therein, the virus replicates and impairs the function of the immune system by infecting macrophages and dendritic cells, as well as inhibiting type 1 interferon responses. These attacks provoke what is known as a cytokine storm. Put simply, a cytokine storm is an overreaction of the host's immune system which leads to death. Given its insidious nature, methods of research have been encumbered by regulations stipulating research on EBOV must be done in a special Biosafety 4 lab. In order to get around this regulation, researchers have developed ways of studying the virus using recombinant single-cycle infectious vesicular stomatitis virus, RSC-VSV. Broad-spectrum antiviral drugs, such as L-like 3-deazinaplanacin or L3-DP, may also provide a way to inhibit Ebola's replication cycle.